Hey all, welcome to Share Trek. This is Raj here. Friends, today grab a beer or your favorite beverage, sit back and try to understand this segment. It's likely to be a bit long, but I will try to explain every concept so that we can develop an understanding of the eOD-GT8 60 MER vaccine. This is an mRNA vaccine aimed at producing broadly neutralizing antibodies or BNABs to tackle multiple strains of HIV. And friends, as I've told you before, I'm not a doctor or a scientist, I'm a genomic investor. And it's just that you liked my HIV related videos, so I have started digging more and more into it. And we have a huge following out here for HIV related information. So I'm doing my best to understand the technologies that are emerging and explain it to you. However, there could be some errors here or there. If I find them, I'll make clarifications. But I try to avoid error as much as possible and use reliable sources. So that's a disclaimer for you right now. And however, before we proceed, I request that you consider joining our Patreon to support this channel and to help continue bringing HIV-specific programming week after week to you. Welcome back, friends. First, I want to explain a few concepts that are a building block for today's story. First, let me explain the exotic name eOD-GT8 60 MER. eOD stands for Engineered Outer Domain, that is eOD, of HIV, GP120. GP120 is the trimer protein that we have spoken about in the past. GP120 and GP41, they form the trimer. And 60 MER stands for 60 engineered copies of HIV's GP120 envelope. MER is where basically the monoclonal antibody. And now we have the HIV's GP120 protein fused to a luminase synthesase protein scaffold. Luminase synthesase basically, is, I understand, is to be B2, vitamin B2 or riboflavin. It's used for cell growth. So I don't know how it works there, but this is my understanding of the title of this particular vaccine. It seems that this vaccine is made on nanoprotein and it's got repeating structures, and that's what I understand overall at a very high level. I'll be reading more on this and I'll come to you with better understanding and simpler explanation as I understand more of this. Next, I want to explain to you another terminology which we need to understand in order to understand this particular vaccine. The terminology is VRC01 class HIV-specific B cells. So VRC01 class HIV-specific B cells are a type of immune cells that play a crucial role in immune response against the human immunodeficiency virus or HIV. To understand these cells better, let's break down the key concepts. B cells are a type of white blood cells involved in the adaptive immune system. They are responsible for producing and releasing antibodies, which are proteins that recognize and bind to the specific pathogens, such as viruses, bacteria, or other foreign substances. These are very high-level definitions. And friends, if you want to have more detailed definition, just go back to my mini-series on human immune systems, where I have spoken about the entire cycle of how macrophages digest pathogens and present the antigen, how naive CD4 T cells pick them up, and how B cells end up getting the same antigen. And when a CD4 T cell with the antigen meets a B cell with the same antigen, then there's a whole cascade that happens where the B cell creates memory B cells and plasma B cells, and the plasma B cells are the ones that create the antibody that can neutralize or attach itself to the antigen that it was carrying in the first place. So the whole thing has been summarized in our definition out here, description out here. So that said, let's look at the next terminology we need to understand, that is HIV-specific B cells. These are B cells that have been activated and specifically recognize components of the HIV virus. When the immune system encounters HIV, some B cells will produce antibodies that can bind to and neutralize the virus. These B cells are known as HIV-specific B cells. So these B cells, when they were naive, they got exposed to HIV-specific antigen, and now they are B cells that are HIV-specific B cells because they are producing antibodies that are specific to HIV. VRC01 class antibodies. This is the other terminology we need to understand. VRC01 is a type of broadly neutralizing antibodies, B and AB. They are a superior type of antibody. VRC01 is a type of broadly neutralizing antibody that was isolated from a HIV-infected individual. These antibodies are capable of neutralizing multiple strains of HIV by binding to a specific site of the virus called the CD4 binding site. The VRC01 is considered a prototype antibody within a larger class of BNABs targeting the CD4 binding site. VRC01 class HIV-specific B cells is the other thing that we need to understand. VRC01 class HIV-specific B cells are B cells that produce antibodies very, very similar to VRC01. These B cells have undergone a maturation process where they develop the ability to produce antibodies that are specifically recognize and bind to CD4 binding site on HIV. This recognition is crucial for neutralizing the virus and preventing its replication. It's important to note that VRC01 class antibodies are being studied for their potential therapeutic and preventative use against HIV. Scientists are investigating ways to harness these antibodies to develop vaccines or antibody-based therapies to combat HIV infection. By understanding the biology of VRC01 class HIV-specific B cells, researchers can gain insights into the immune response to HIV and explore strategies for enhancing antibody response against these viruses. One such initiative following this line of research is the team from Fred Hutchinson Cancer Center and Scripps Research, who with other collaborating institutions have been working on this concept. Recent news from this team highlights that in the IAVI G001 phase 1 clinical trial, they tested a therapy for safety and immune response successfully using a self-assembling nanoparticle HIV vaccine. You will recollect from my earlier videos that helper T cells with specific antigen meets B cells with the same antigen and start to cascade, where B cells create plasma B cells and memory B cells. The plasma B cells then produce the antibody for the target antigen common to the B cell and CD4 T cell. In this case, the Scripps research team identified an antigen which, when introduced into the cycle, converts naive CD4 and B cells into activated CD4 and B cells, stimulates VRC1 class B cells. The team says it was quite impressed with the vaccine candidate as it produced a tremendous T cell response in most of the trial participants. At present, the team feels that booster vaccines may be needed to increase the volume of VRC01 broadly neutralizing antibodies that are able to neutralize almost 90% of known HIV strains. The 7 key findings from these studies include: number 1, vaccine-specific CD4 T cells were introduced in almost all vaccine recipients. So that's a very, very important aspect, which means that most people were sensitive to this stimulus and they reacted the way they should for this vaccine to succeed. Second point was that lymph node GCT follicular helper cells increased after vaccination. Let me explain this concept of lymph node GCT follicular helper cells before we look at the other findings. Lymph node GCT follicular helper cells, also known as TFH cells, are a specific subset of T cells that play a crucial role in immune response within the lymph nodes, particularly in the germinal centers or GCs. To understand these cells better, let us break down the key concepts. Lymph nodes are small bean-shaped organs distributed throughout the body and are a part of the lymphatic system. They act as filters for the lymph fluid and serve as sites for immune cell activation and interaction. Germinal centers, or GCs, are specialized regions within the lymph nodes where B cells undergo intense proliferation, selection, and maturation. GCs are important for generating high-affinity antibodies during an immune response, such as in the context of an infection or vaccination. T follicular helper cells, or TFH cells, are a specific subset of CD4 T cells, also known as helper T cells, that are primarily located in the germinal centers of lymph node. They have a unique molecular and functional profile that enables them to provide critical help to B cells during antibody production. TFH B cells interaction is another thing that we need to understand. Within the germinal centers, TFH cells interact closely with B cells, providing signals and guidance necessary for B cell activation and antibody production. TFH cells express a protein called CXCR5, which helps them migrate into the B cell follicles where the germinal center form. Once inside the germinal centers, the cells provide several important signals to B cells, including the expression of molecules like CD4L or CD4 ligand and cytokines such as IL-21, which promote B cell proliferation, antibody class switching, and development of high affinity antibodies. GCTFH cells is another concept we have to understand. GCTHF cells specifically refer to TFH cells found within the germinal centers. These cells have undergone a differentiation process within the lymph nodes and acquire specialized features that make them particularly efficient in providing help to B cells within the GC environment. GCTFH cells are critical for the development of effective antibody responses against infections and vaccinations. They contribute to the selection of B cells with high affinity antibodies, assist in the process of affinity maturation, and promote the differentiation of B cells into long-lived antibody-producing plasma cells or memory B cells. Understanding the biology and function of GCTFH cells is important for studying immune responses, developing vaccines, and designing therapeutic strategies targeting antibody-mediated immunity. So that was the concept that I wanted you to understand around GCTFH cells. The next point of learning from the phase 1 trial was lumazine synthase protein needed for self-assembly of particle also induced T cell responses that can provide additional help to ultimately enhance efficiency in a sequential vaccine strategy. Vaccine-specific CD4 cells were polyfunctional and had diverse phenotypes. Let me explain this other finding. Let me briefly explain polyfunctional CD4 T cells. Traditionally, CD4 plus T cells have been classified into distinct functional subsets based on cytokines they produce and the immune responses they promote. For example, Th1 cells primarily produce interferon gamma and are involved in cellular immune responses against intercellular pathogens, while Th2 cells produce interleukin 4 or IL-4 and IL-13 and are associated with antibody-mediated immune responses against extracellular parasites. However, recent studies have revealed that there is a CD4 plus T cell category that can exhibit functional diversity and flexibility. Polyfunctional CD4 plus T cells are characterized by their ability to produce multiple cytokines and exert multiple effector functions simultaneously or sequentially. These effector functions can include the production of cytokines such as IFN, IL-2, IL-4, and IL-17, or tumor necrosis factor alpha, that is called as TNF alpha, as well as the expression of surface molecules associated with cytotoxicity or immune activation. The presence of polyfunctional CD4 plus T cells is often associated with a robust effective immune response. These cells are believed to provide a more comprehensive and coordinated defense against pathogens by simultaneously or sequentially engaging different aspects of the immune system. And polyfunctional CD4 plus T cells are commonly observed in various infectious diseases, including viral, bacterial, and parasitic infections, as well as in autoimmune disorders and cancer. So these are some kind of super CD4 T cells. Assessing the polyfunctional profile of CD4 plus T cells through techniques like flow cytometry or cytokine secretion assays have become an important tool in the immunological research. Understanding the characteristics and functions of polyfunctional CD4 plus T cells can provide insights into human immune response, vaccine development, and the development of therapeutic strategies targeting specific disease. And then we have this lumpsin specific CD8 T cells, which were highly polyfunctional and had a predominantly effector memory phenotype. So this is another finding that they had. I suspect the term lumpsin specific CD8 T cell is a hypothetical and used here as an illustration. In reality, CD8 T cells are typically specific to antigen derived from pathogens such as viruses, bacteria, or tumor. The activation and expansion of antigen-specific CD8 plus T cells play a crucial role in controlling infections and eliminating abnormal or infected cells in blood. So I need to study this a little bit more to understand and have better clarity. So CD4 T cell responses were also found to be driven by immunodominant epitopes with diverse and promiscuous HLA restriction. Let me unpack this concept as well. The concept of immunodominant epitopes with diverse and promiscuous HLA, which is human leukocyte antigen restriction, refers to specific regions known as epitopes within proteins or antigens that elicit a strong immune response and are recognized by a diverse range of HLA molecules. Epitopes are small specific regions within proteins or antigens that are recognized by the immune system, particularly by T cells. T cells recognize antigens through the interaction of their T cell receptors, TCRs, with peptides presented by HLA molecules. HLA molecules are also known as human leukocyte antigens or major histocompatibility complex or MHC molecules, are proteins present on the surface of cells. They play a critical role in presenting antigenic peptides to T cells, allowing the immune system to recognize and respond to foreign substances. Immunodominant epitopes within a given protein or antigen. There are certain epitopes that have a higher capacity to elicit an immune response compared to others. These epitopes are referred to as immunodominant epitopes. Immunodominant epitopes are often primary targets of the immune response and play a significant role in shaping the overall immune response to a pathogen or antigen. And then we have to understand diverse HLA restriction. HLA molecules are highly polymorphic, meaning they exist in multiple variants or alleles within the human population. Each HLA variant can bind and present different peptide antigens. Diverse HLA restrictions refers to the fact that different individuals may express different HLA variants, leading to variations in the epitopes that can be presented to T cells. Promiscuous HLA restriction means that some HLA molecules have the ability to bind and present a wide range of different peptides regardless of their amino acid sequence. These HLA molecules are referred to as promiscuous because HLA restriction allows a single HLA variant to present a broad range of epitopes, increasing the chances of T cell recognition and immune response. When immunodominant epitopes exhibit diverse and promiscuous HLA restriction, it means that these epitopes are capable of stimulating a robust immune response in a diverse range of individuals with different HLA variants. This is advantageous for vaccine development and immune responses to pathogens because it ensures that a significant proportion of the population can mount an effective immune response against the immunodominant epitopes regardless of their HLA genetic background. Finally, CD8 T cell responses to lumpsin were driven by HLA A02 restricted immunodominant epitopes. B and T cell responses correlated with but not between LN and peripheral blood compartments. So that was the last finding. I do not yet understand the last finding. I'm still studying up on that, and I need to learn more building blocks of knowledge before I can comprehend the importance of most of the 7 findings of this study. But I have tried to present however much I could understand. And just to summarize this whole thing, the way I would characterize this is, let us take the example of COVID-19. COVID-19 had an antigen. So the mRNA vaccine produced the antigen within our body, and that helped our body to go through this whole process whereby the CD cell, a naive CD cell, becomes activated and creates helper T cell, which in turn goes and finds a B cell which has the similar antigen and tells the B cell to produce antibodies for the COVID-19 antigen. So this was the cycle, and it was a very simple antigen. When it comes to HIV has got a lot of mutations and it's a complex virus. And because of its various mutations, having a single antigen is not going to help. So we need a complex antibody, and that complex antibody is the broadly neutralizing antibody. So the whole effort in this particular vaccine, which is called eOD-GT8 60MER, is basically to create a vaccine where instead of an antibody, we have broadly neutralizing antibodies. And in order to create that, it's using a much more effective antigen. So that's how I understand this particular vaccine. And the fact that it has produced impact on almost 90% of the known HIV strains is, number one, a very, very big achievement. Number two, the fact that most of the people on whom this vaccine was tried gave an immune response as expected, shows great promise. I have reached out to the Fred Hutchison Cancer Center by email to the coordinator to find out if they can give me some information about when phase 2 clinical trial will start, and also if any one of their scientists from their research team can join us on a Zoom interview so that we could understand this better. So I'm looking forward to hearing from them, and if I do hear from them, I'll keep you updated, and hopefully we have an interesting interview and we understand this much better. I'm going to continue to read up on this particular vaccine and develop a better understanding. I wish I had somebody who could do animations, because this concept could be much easily understood with animations. When I was trying to understand this, I had a sheet of paper in which I was drawing in order to understand the concepts, and I've broken them down into building blocks. You might have to listen to this video more than once in order to understand it. I've tried to experiment with trying to provide more detailed information in this video. I would like you to give me comments letting me know if this is too much information. If you want just simplified content, please put that in the comments below so that I can calibrate my approach. With that, I'd like to end this video. Thanks and have a great day. Bye for now.